Hello, today we're going to be looking at paper chromatography of chlorophyll. The first thing we're going to do is prepare our chromatography paper. Now we only ever handle our chromatography paper by the edges because we don't want fingerprints or dirt or grease to impede or affect the flow of the solvent and the material up the paper. The first thing we're going to do is very carefully, without putting our fingers all over it, measure 1 centimetre up from the bottom of our chromatography paper. Now this is the line, this is the point at which we're going to put our samples. Okay, now we need it clearly marked so that we can make sure that we know where the start line is for our samples and that all our samples are starting at the same point if we were doing a comparison. So for example, if we were comparing two separate leaves, we'd want to make sure that they start and finish at the same point, so then we can calculate what's called the RF value. So when we've done that, okay, we can put that aside. Okay, we're going to extract what we're going to run. So in this case, we're going to be doing chlorophyll. Okay, now chlorophyll is the little green. It's the little green chemical within the chloroplasts of our leaves, okay, and we see it as green, but it's actually made up of several different colours, and that's what we're hoping to see, is that we're hoping that we will see different colours spread out as we run our chromatography. So we're going to put some spinach into a pestle and mortar, okay, and a small amount, and I do mean a small amount, of sand. Now the sand is purely there to act as an abrasive, okay, so that when we are grinding with our pestle and mortar, it's helping us mush up our spinach. Now we want the spinach to be well and truly broken. Okay? We need to get the walls, the cell walls broken down so that we can then get a reasonable amount of chlorophyll out from within those cells. If we do not break the cells down well, we will not have a sufficient material in our sample. Now we need to do this in a way that we don't get it absolutely everywhere. Okay, now chlorophyll is green and it will stain, so it will stain your clothes and your fingers if you get it all over them. Okay, now we're going to add a small amount of propanone, okay, known as acetone, okay, to our chlorophyll. Now we do mean a small amount, you're only talking a mil or two, okay, that's going to act as our extraction solvent. Okay, so we're going to get that is going to grab hold of the chlorophyll and turn it into a liquid. And it's important that our extraction solvent is something that will evaporate, because we don't want our extraction solvent to stay on the paper. So when we've transferred our material, the acetone will evaporate away and leave us with a dry sample of chlorophyll on our chromatography paper. Now we're going to use, okay, a capillary tube. Okay. Now all I've done is put the capillary tube in to pick up some of the liquid. Hopefully you can see that there's a nice little bit of green liquid in the bottom of that capillary tube. Okay, now to pick up, we put it in, we put our finger over, we put it in, we put our finger on and we remove the capillary tube. Now in this case I'm going to do 2 spots, okay, about 1/3 of the way into the paper on our line is the first spot. And I'm going to do a second spot 2/3 of the way across on our line. Now, the more spots of this you do, the more concentrated the sample you get. Okay? If you only do 1 spot, you're going to have quite a weak sample. So what we would do is we would put our spot on, we would leave it for a few seconds to dry, and it's nearly almost completely dry. Okay, I'll show you our little chromatogram. Okay, so we have our nice 2 green dots. Okay, so it's nearly almost completely dry. Then we're going to repeat the process. Now we would do this normally, okay, about between 5 and 10 times, depending upon how strong our sample was. Okay. Now every time we're spotting one, we're increasing the concentration. The more concentration of the chlorophyll of the material we have on the paper, once we separate it out, the clearer the bands will be. If you don't get enough of the material on there, you might not be able to see the bands. Okay, once we've done that, and we've repeated it about sort of 10 times, okay, leaving it to dry each time in between, we're going to leave it to dry for the final time. While we're leaving it to dry, we're going to set up our last bit.
Water.
Okay, so we're going to use an extraction solvent. So in this case, it's a mix of acetone that we used earlier and petroleum ether, okay, which is a makeup of a fraction of petroleum. So it's a 70% mix of petroleum ether to 30% propanone. Okay, now we're going to put it in. Now obviously you would normally do this in a fume cupboard, but there's only me here, so. Okay, now we want the solution to be below the level that we're going to put our, below the level of our chromatography samples when we put the paper in. So if it's more than 1 centimetre, okay, or the samples are in the solution, they will come out into the liquid. So it needs to be below the line and below the samples. Okay, so if you accidentally put too much in, just take a little bit out. So you need about, we've done a centimetre line, so you need about a 5 ml, half a centimetre depth of solvent in the bottom. When we've done that, we put our sample in, okay, sample down, making sure that the sample doesn't go below the level of the liquid, and we put a lid on to stop any vapours getting out, because we want the solution to run up the chromatography paper. If there's no lid on, it will come out into the air and dry out in the air before it gets all the way up. We're then going to leave this to run, okay, as you may or may not be able to see. Okay, the solvent is already starting to move its way up the paper. Now we want it to get to a couple of centimetres before the top of the paper. Okay, so we do need to keep a close eye on it. It will probably take about 20 to 30 minutes for this to run. So I'm not going to keep you here for 20 to 30 minutes. I'm going to explain what we're going to do next. So when we take it out, okay, when we take our chromatography paper out, you will have a line of liquid where it hasn't dried. Okay, that's called the solvent front. Okay, and so we'd have the start line and we have where the solvent's got to. Now obviously if we were to just leave it as is, the solvent would dry and you wouldn't be able to see that line. So what we do is when we take it out, we quickly draw a line across with a pencil where the solvent has got to. And then knowing the distance that your sample has travelled and the distance from the start to the solvent front, okay, here we go, I've got one that we prepared earlier, you'll be able to work out your RF value. So if you look at this one, okay, if you look at this one, I don't know how clearly you can see it, but we had the start line with the sample, we've got very faint samples on here, and we have a pencil line across the top which marks where the solvent front got to. Okay, and that's it. Thank you very much.